Good afternoon. This is Dr. Dan Guerra of VerEvMed in the heart of VerEvMed Studios in the heart of the Pacific Northwest. Today, a rather balmy day, well above freezing, making me think about how spring is just around the corner. I mean, only 3 months away. I want to embark on a sphingolipid discussion that works in many ways in complete opposite as did our friend ceramide. Remember, ceramide is that sphingolipid which is associated with programmed cell death, and it was the topic of the talk I just gave. Today, we're going to talk about sphingosine 1-phosphate signaling. And so rather than being involved in deep winter, I chose a summer figure from a recent trip to Utah and summer images to make you think about the fact that all things change. And so even though it might be dark and dreary wherever you are, soon it will be blue skies and warm weather and all the things that accompany that wonderful time of year, particularly the spring when things start coming out, flowers and such, and birds coming back. All right, so with those happy thoughts, let's get started. Again, this is VerEvMed. VerEvMed is a company that is involved in consultation in biomedicine, as well as in various prognostications of molecular sciences, using the primary literature, the published literature you find in journals, scientific journals, that is, and then bringing forth and vetting that evidence and discussing it, comparing it according to the foundationalism and the other relative associations of the current literature with literature we know that is generally accepted, knowing that science is research and research always changes. So I'm publishing this particular talk in January 2018. So all the things that we talk about today will be up to this point. Now, in 2 weeks, things could change because publications keep on coming out. So this is my Facebook page down here. There's my private email that you can contact me directly to. And here are our email and website for the company. All right. Now, start right in with the paper. Paper was published now a few years back in a journal called Development. Basically, what it's looking at is this. It tells you that sphingolipids are linked to membrane function and transduction mediated responses. And it further describes in this one paper that a key polar molecular species, which is, as you might remember from previous talks of sphingolipids, is relatively rare because they're very nonpolar compounds, even for lipids. A key molecular species that is somewhat polar is sphingosine 1-phosphate. I mean, it's going to have different chemical properties, and therefore it's going to move through membranes, and it's going to function intracellularly different than ceramide, right? Just because of its chemistry. So sphingosine 1-phosphate, or S1P, is found in the extracytoplasmic space, where it binds to a group of cell surface receptors, thus regulating migration, differentiation, and survival. So S1P is one of those lipids that can leave the cell and re-enter. Okay. S1P is involved in migration, cell induction, and even in biomechanical signaling. That signaling is essential in development, angiogenesis, cardiogenesis, limb development, and indeed neurogenesis. So, a couple of diagrams showing you the basic molecular architecture and motif for sphingosine 1-phosphate. You can get sphingosine 1-phosphate on various cell types there's in circulation, platelets, mast cells, monocytes, etc. The sphingosine 1-phosphate can be secreted from these cells, can move through any number of these receptors. Once it moves through those receptors as a ligand, it's going to cause intracellular events. Those intracellular events are usually associated with various G proteins. Those G proteins, in turn, all functioning against the plasma membrane initially, are associated with the effector molecules in that cell. So we have kinases, the P13 kinase, RAC. We have adenylate cyclase. We have the ERK kinase, phospholipase C, and some of the players I know we've talked many times about, Rho, JNK, and ERK, kinases that are associated with cell proliferation, cell cycle arrest, and cell cycle movement, depending on their native and inbound cytoplasmic environment. Then you get a bunch of cellular responses. You can see it's a typical signal transduction cascade. Here, a little bit more discrete. Here is where you see it again, reacting with G proteins. The receptor, the sphingosine 1-phosphate receptor, is the canonical one found in the membrane. And you can see that it's involved in apoptosis survival. Okay, so rather than inducing apoptosis, it's a mechanism to survive programmed cell death. It's involved in cellular movement or chemotaxis, migration of cells and embedding, and indeed if it's a cancer cell, metastasis. Calcium influx, really important in energizing and altering the cell fate and whatever happens metabolically in a cell that has calcium influx. And also, very importantly, and again, antithetical to ceramide, is how sphingosine 1-phosphate is involved in proliferation and differentiation, you see? Okay. So here we're looking at it in various cell types, endothelial cells, inflammatory cells, or indeed in cancer cells. Again, just like all signaling molecules, the flavor of the receptor, which is associated with the type of cell that's expressing the receptor, is going to dictate in many ways what the ligand binding will cause intracellularly, right? So if you're a liver cell, you're not going to you're not going to have angiogenic or lymphanogenic properties, right? So, but if you're an endothelial cell and sphingosine 1-phosphate binds to its receptor, S1PR, shown up top here, that can induce angiogenesis and lymphangiogenosis, okay? It can generate new blood vascular system, both in the lymphatic and in the regular circulation. An inflammatory cell we'll talk about later, but depending on the inflammatory cell, whether or not it's a macrophage or a T cell, for example, or a B cell, sphingosine 1-phosphate is going to have multiple pleiotropic effects. So the cell environment plays a role, and the receptor that's expressed on that cell also plays a role, obviously, in the signal transduction cascade. Here in a cancer cell, you can see that you can synthesize sphingosine 1-phosphate using this kinase. It happens in the nucleus. So from sphingomyelin to sphingosine 1-phosphate, that's one way of doing it. From sphingosine, not sphingomyelin. I was thinking ahead there a bit. There's a receptor binding to it, right? And remember, we told you in cancer cells, proliferation, migration, survival, so it's a bad player if you want to look at it that way. And as we also mentioned, sphingosine 1-phosphate can leave the cell and can travel outside to different cell types, right? So it is indeed a paracrine hormone, okay? All right. So let's take a look at this embedded within our understanding of sphingolipid metabolism. Now, again, remember, we haven't even scratched the surface of how sphingolipids are involved in membrane activity, right? We're just looking at it as a signaling molecule. But I also want you to keep a really important mindset on the fact that sphingolipids are really important, not so much in bioenergetics, but in signaling cell fate. Now, phosphoglycerolipids do the same thing, and they have a lot of airtime, a lot of print, a lot of ink has been made on phosphoglycerolipids, such as the phosphatidylenosyl phosphates, for example. I'm sure most of you medical people have heard about that. A lot of you graduate students, et cetera, heard about that. But the sphingolipids are relatively new on the scene because their full gambit of cellular activity is only now being explored at the full level because of the paradigm shifts we've had in cancer and cardiovascular disease research, for example. At any rate, look here. So remember that you make ceramide multiple ways. Denovo Synthesis. You can also break down sphingomyelin via the sphingomyelinase, right? You can resynthesize it. Remember that? Couple that there. Remember that ceramide, its main claim to fame is that it's involved in programmed cell death, that is apoptosis, cell cycle arrest, and cellular senescence, okay? Things that shut down cells, something that you would definitely want in a cancer cell. Now, ceramide can also be metabolized to ceramide 1-phosphate. Now, that tends to work 180 to the parent compound ceramide. Remember, there's a lot of chemical change here. This is a much more polar molecule, adding that phosphate ester. So that's one thing. The physical chemistry of the compound plays a role in where it traffics and how it binds and its concentration, biological activity at what concentration, those kinds of phenomenon, right? Straight order chemical kinetics. Okay, so there is this couple here, and we'll talk later about ceramide 1-phosphate. But most importantly, today I want to emphasize sphingosine 1-phosphate. See here? So Remember that you break down ceramide via ceramidase. You remove that amide-linked fatty acid. You get a sphingosine base, which can be phosphorylated by a kinase to sphingosine 1-phosphate, which can be broken down, and there's no coming back after this, to ethanolamine 1-phosphate and palmitaldehyde. Now, S1P is involved in cell motility, as we just said, survival rather than death, proliferation, right, rather than arrest and senescence, and indeed, if it's an immune cell type of situation, environment, you're going to have inflammation. Okay, and remember too that you can make sphingosine 1-phosphate and build back sphingosine, and sphingosine can build back ceramide. So this is a really interesting couple because you can go in multiple valencies, right? Ceramide to sphingosine, which has very little biological activity as far as we know, but don't hold your breath because sphingosine might actually play a role in its own right as research goes on. But notice how ceramide is intimately linked to S1P, and ceramide is also linked to C1P, and any of these can be interconverted depending on the enzymes involved, the relative substrate concentrations, and whatever the upstream and downstream signaling is. It's going to alter and change the metabolic flux in these multiple intermediates. Also remember that the membrane lipid sphingomyelin is kind of a parent compound to all of this. And also Denovo Synthesis plays, of course, a super crucial role. So let's go back. Sphingomyelin generates ceramide, which is further processed to sphingosine by the enzyme ceramidase. Sphingosine is then phosphorylated by any one of a number of sphingosine kinases to give rise to S1P. Now, when I say any number of isozymes, that's really critical, really important in cell type, tissue type, and also whatever signaling or stress phenomena is going on, pathophysiological or straight up immune response, stress response. And those isoforms are going to play key roles in S1P signaling. S1P concentration is regulated, therefore, via biosynthetic enzymes and degradated ones, as we just saw in the previous slide. Now, what's really interesting, and you also saw this so far, S1P can be exported by specific transporters, and that secretion also can occur of the enzyme itself. So you can get sphingosine 1-phosphate synthesized in the extracellular matrix. Yeah, pretty complicated stuff, right? But if you remember, sphingomyelinase also gets exported. So you've got a lot of monkey business going on with sphingolipids, not just what's going on in the cell, but they tend to want to bring their enzymes with them. When they bring their enzymes with them, you can alter cell fate, and not just that, signaling throughout a tissue mass or even the whole organism. So that's why sphingolipids are so exciting and interesting, because they seem to have a very significant and potent effect that is extremely complicated. And complicated things are always more fun than simple things. Okay, so blood plasma contains high levels of sphingosine 1-phosphate relatively compared to a lot of other lipids. That's 1 micromolar. That's pretty high, actually, for one particular lipid. Interstitial fluid levels of S1P, however, are low. So that means that the S1P is going to move from the blood into the cell, right? Just because of osmosis. That's how it works, right? So that's going to generate, as I'm saying here, a gradient, driving S1P function into the cell. So what about the receptors? Basically, the sphingosine 1-phosphate receptors are intimately linked to GPCRs, G protein coupled receptors. And therefore, they're related in many ways to other signaling pathways we've talked about here at VerEvMed. Lysophosphatidic acid, phosphatidic acid, other phospholipids, as well as cannabinoid receptors, all utilize a similar system. Most cells actually express one or more subtypes of these receptors, and the result is the activation of a whole family of small GTPases, such as Rho, RAC, and RAS, which again I've talked about at great length in previous lectures, which all can be observed, by the way, on my Facebook. Downstream effectors of sphingosine 1-phosphate receptors include AC, adenylate cyclase, that makes cyclic AMP, and a really important second messenger involved in metabolic interactions and phosphorylation of cyclic AMP-dependent protein kinase activity substrates. P13 kinase, phospholipase C, protein kinase C, and intracellular calcium. So those are major intracellular signaling phenomena, right? When you write down those 4 or 5, you're talking 50, 60, 70% of everything that goes down in the cell that's exciting and interesting and dynamic. Transcriptional responses, of course, you have with Rho GTPases, and that's, of course, as we're saying, coupled with the sphingosine 1-phosphate receptor. It can also activate another whole pathway, which I talked probably now a few months back on, the hippo signaling pathway. and its associated transcriptional effectors and histone deacetylase. Now, that's all nuclear activity, which means you're going to be involved in, chromatin remodeling, and the chromatin remodeling ultimately can lead to alterations in gene expression at the transcriptional level. There's also some suggestion that sphingosine 1-phosphate and indeed the other sphingolipids I've been talking about may play a role in recombination, DNA recombination, DNA repair. So we're not through it all by any means just talking about signal transduction outside the nucleus. That's what I'm trying to get at here. All right. So basically here you've got tyrosine kinases here, receptor tyrosine kinases, working to phosphorylate a series of other kinases. We talked about these many times, as you recall. Here's your kind of canonical pathway going through the AKT, the TSC, and then, of course, the mTORC pathway, the target of rapamycin complex. All of this ultimately can lead to basically macromolecule production, right? And that, because you're making a lot of proteins, for example, then you're going to be turning on cell cycle and get cell growth, and you're going to get increases in organ size. For example, adipose tissue comes to mind, that organ, adipose. G protein couple receptors working through the hippo pathway, work through a couple of other systems, other enzyme complexes. They also work through an interfering RNA species called miR29, which blocks P10. That is a phosphatase. So this whole pathway is just reinforcing the RTK pathway. I'm just here just giving you dynamics of like what receptors do, because it blocks the P10. If you block the dephosphorylation, you're going to drive the positive effect, right? And that's what's going on. So cell proliferation, cell growth, organ size, all are enhanced by these pathways. And sphingosine 1-phosphate's involved in this. Okay. So what are the major sphingosine 1-phosphate effector systems? That means downstream. Regulation of cell-cell and cell matrix adhesion is really important for S1P. Signaling in the endoderm and in heart development and repair is also really important in S1P. So cardiovascular health and also cardiovascular disease is intimately linked to S1P. Indeed it is. And control of cell trafficking in and out of the vascular system is also intimately involved. So we can also talk about atherosclerosis and various aspects of, again, cardiomyopathy and cardiohypertrophy. Okay, all of those things are going to be linked with sphingolipid metabolism. What about the immune responses? Okay, those are organ responses, immune responses. Because of the sphingosine 1-phosphate gradient, it regulates trafficking of immune cells from lymphoid into circulation. Cell surface lipid phosphate phosphatases degrade sphingosine 1-phosphate. Sphingosine, of course, you can remove that phosphate. Once you do that, basically your signaling is done with, okay? Although there is a kinase that can add it back. And as I said, the kinase is out there in extracellular matrix, out there outside the cell. The S1P transporter, called SPNS2, expressed primarily in the lymphatic endothelial cells, is required for efficient egress when you're talking about immune transport and mobility. And the uptake and metabolism of sphingosine 1-phosphate from circulation by endothelial cells may be involved in lymphocytic trafficking. And of course, the receptor of cell surface lymphocytes is crucial for this egress. What about neurogenesis? Yep, sphingosine 1-phosphate is involved there. Receptors are enriched in neuronal tissue, so that usually suggests it's sphingosine 1-phosphate like as a ligand there, altering, activating perhaps, or otherwise modifying neuronal activity. Mice lacking, in fact, sphingosine kinase isozymes exhibit severely disturbed neurogenesis, so obviously they are important. S1P treatment of neonatal brain results in cellular movement leading to the formation of neural folds such as sulci and gyri. So it looks like neurodevelopment is linked positively with S1P. It's not surprising because it's involved in cell proliferation and also cell morphogenesis, right, development. S1P signaling has been implicated in neuroprogenitor cell recruitment, and receptor targeting reduces neuroinflammation and brain volume loss in humans. Okay, so there's a lot to be said about S1P in the brain, central nervous system, as well as the peripheral too, though. This is a good overview slide talking about here thymus, the non-inflamed lymph node and the inflamed lymph node. You can see here that the sphingosine 1-phosphate receptor on the thymocyte will then cause the movement of the SP thymocyte moving along the pathway here. You have an internalized sphingosine 1-phosphate receptor associated with the CD4 intramembranous cell receptor mediator. Once that occurs, you get this uptake via into this perivascular cell, and this is going to induce an entirely new activity because of the sphingosine 1-phosphate effect on cellular metabolism there. In non-inflamed lymph nodes, you've got a T cell here with nothing going on, right? Very low sphingosine 1-phosphate concentration outside, very high inside, but basically just an internalized sphingosine 1-phosphate receptor. However, when you inflame that lymph node, time for the glasses here, and inflamed lymph nodes activate the T cells, upregulate a couple of cell surface proteins, CD69 and another CD we talked about up here, CD4, and this promotes sphingosine 1-phosphate receptor degradation. Okay, so then here in this activated T cell, you get sphingosine 1-phosphate receptor internalized and degraded. Okay. That's going to cause an association with a dendritic cell via the T cell receptor. So the newly generated effector cells that are going to come off of this degradation of the sphingosine 1-phosphate receptor are going to be effector T cells. So you got newly generated effector T cells. They lose a certain chemokine receptor, CCR7, and they upregulate now nascent sphingosine 1-phosphate receptor, and now they're going in again into circulation, and those effector T cells are entering and ready to respond. You see, they're responding now to circulating sphingosine 1-phosphate because you've charged them with the receptor on the surface because of this modification here, which is associated with inflammation. Okay. So, sphingosine 1-phosphate signaling, off with the glasses, recently targeted in remitting relapsing MS. Okay, yet another disease system that's been looked at. Indeed, sphingosine analog called fingolimod is approved for the treatment of MS, right? Okay, so it's an analog, which means it's going to act as probably an agonist. Might be an antagonist, depending on the concentration. Need to look at the literature. So fingolimod is phosphorylated by the kinase, and it acts initially as an agonist of all those receptors. What did I tell you, right? You got to think about concentration. However, and also temporal activity. How long is it there, this compound, fingolimod? Remember, it's phosphorylated. It's going to touch all of those receptors and activate them, right? All those sphingosine 1-phosphate receptors. Ultimately, that compound will function as an antagonist, as binding to the receptor causes downregulation, which has always happened. You overstimulate a receptor, you downregulate, you pull it out of the membrane, and you degrade it via endocytosis of that receptor. And that's exactly what happens in lymphoid organs. So the inhibition of lymphocyte trafficking in the CNS may be a major mechanism by which this compound achieves therapeutic efficacy. So remember, with multiple sclerosis, you have this inflammation, right? And so that inflammation ultimately leads to sclerosis of the neural system. And if you inhibit lymphocyte tracking into the CNS, you're going to knock all that back. You're going to cause a blockade. So that's very likely the mode of action of fingolimod. Phosphorylation, acting first as an agonist and an antagonist for the sphingosine 1-phosphate receptors in lymphoid organs. All right. Next paper here that we're going to talk about was published about 3.5 years ago. Sphingosine 1-phosphate prevents chemotherapy-induced human primordial follicle death. Now check this out. This is a reproductive paper. That is, it doesn't reproduce itself. That means it's about human reproduction. So cancer drugs have non-specific effects on healthy tissue, including gonadotoxicity. So it's an unfortunate but well-described phenomenon that when young children, very sadly, very unfortunately, have multiple forms of cancer, which can be, of course, high morbidity and, of course, mortality. And they're given chemotherapeutics, your classical chemotherapeutics. Those cancer drugs have tremendously bad side effects on development of those young children. Just one of the side effects is gonadotoxicity. It sterilizes them. Real problem. Real sad and real scary. So if we know why that happens, right at the signal transduction level, at the biochemical level, then maybe we can create better drugs that don't target the gonads and cause sterility. So that's just one argument there, right? So, all right. So the mass of primordial follicles present in each ovary is irreplaceable. You know this probably from basic biology, right? So a depletion induced by chemotherapy at a young age leads to premature ovarian failure and, as I've just said, infertility in women. Reproductive concerns account for about 72% of the variance in scoring and the quality of life. So this is really important to us. And to address the issue, embryo and oocyte cryopreservation are now considered established methods, and of course in vitro fertilization success rates have caught up with in vitro fertilization techniques, right? So in fact, I know this firsthand. So cryopreservation of oocytes and embryos may resolve the issue of chemotherapy-induced infertility, right? But they do not address the issue of impaired ovarian endocrine function that's still there. So autotransplantation of cryopreserved ovarian tissue can address those reproductive endocrine complications that are otherwise derived from, as a result, or consequent to chemotherapy. Okay, so there's multiple avenues to take, including surgery. So, what are the pharmacotherapeutic alternatives? The authors of the paper we're looking at here promote a non-invasive, non-surgical, right, pharmacotherapy to block the apoptotic effects of chemotherapeutics on ovarian follicles and therefore to preserve ovarian function. What's the information we need to know to be able to evaluate this? First of all, cyclophosphamide is an alkylating agent, very important, very toxic, very nasty, but very useful chemotherapeutic. And the way it works, mode of action, is induces cytotoxicity by cross-linking DNA. Okay, and then when that cross-linking occurs, it induces DNA breaks, and those breaks then do not get repaired directly or correctly, and you get multiple mutations, and that kills the cancer cell. Doxorubicin is an intercalating agent, so it works a little bit differently. It doesn't alkylate, it just intercalates because of the structure of it. In fact, it acts like a nucleotide in a way. And that causes DNA damage via strand insertion, right? So you get triple helices, and you get, again, mutations caused by that. Recombination, repair, and replication are interrupted because of doxorubicin. Both of those drugs, cyclophosphamide and doxorubicin, are common chemotherapeutics today, for sure, still. Yet they cause human primordial follicle apoptosis and ovarian reserve depletion. So that's the sad part of them, okay? And they're used on young people, for example. Sphingosine 1-phosphate, though, is known, and you would know this too if you just saw my last two talks, inhibits ceramide-induced cell death and apoptosis. So, we've got a phospholipid here, and that is a phospholipid, by the way. It happens to be a sphingosine phospholipid, not a glycerol-containing phospholipid. And it seems to work counter to ceramide. So maybe if we promote sphingosine 1-phosphate, we can block this cell death. Blocking this cell death then could preserve fertility, right? So S1P appears to decrease radiation and chemotherapy damage too. So wow, that is fantastic. Not just chemotherapy damage, but also damage caused by radiation and as we already said, chemotherapy. Okay. So just to remind everyone, I know you've seen these pathways multiple times now. I'm just bringing it back in this particular talk. Remember, the Denovo synthesis of the sphingolipid starts with palmitoyl-CoA and L-serine, and then a very interesting series of reactions that lead ultimately to ceramide, which contains an amide-linked fatty acid, to the nitrogen atom that originally was donated by L-serine. Certain drugs here that block ceramide metabolism, like tamoxifen and 1-phenyl-2-palmitoylamino-morpholino-1-propanol, or PPMP, known affectionately as thereof, blocks the metabolism of ceramide. This is a glycosphingolipid synthase, right, or a glucosyl ceramide synthase, right, adding sugars, making those glycosphingolipids. It blocks that pathway, it blocks the further anabolism of ceramide, and do so, it builds up ceramide, right? Remember, when ceramide builds up, it works through the junk kinase pathway. There's an intermediary to get in the mitochondria called BIM, kind of an adapter protein system. Once it gets into the mitochondria, remember, it kicks out the cytochrome C in the outer membrane. It turns on the SMAC Diablo pathway, and boom, mitochondrial-associated programmed cell death, also known as apoptosis. And here's a little insert just showing you how all of this is embedded in this, again, metabolic network interaction and reversal. So here's your de novo pathway down to ceramide. Ceramide broken down to sphingosine. Sphingosine is sphingosine 1-phosphate. But look at how some of these arrows are reversible like this one here. Very important, okay? So you can rebuild ceramide, okay? So, and remember that ceramide also, again, is involved in the synthesis and comes from the degradation of complex sphingolipids that are membrane associated, for example, in the central nervous system. All right, back with the glasses. So let's take a look here at S1P metabolism. So start off, here's the plasma membrane. Here's the entire Golgi apparatus. You have sphingomyelin being synthesized from ceramide. You have ceramide being used to make glucosyl ceramide. And also there's a reaction called glucosyl sphingolipid lyase, which re-synthesizes the glucosyl sphingolipid pathway, okay? And there's a transporting mechanism called the glucosyl ceramide transport, or FAPP2. All that's going on in the Golgi. So a lot of interaction at the ceramide sphingomyelin level and the glycosphingolipid metabolism level. Now, in multiple compartments, you have these pathways going. Sphingosine going to sphingosine 1-phosphate, sphingosine 1-phosphate going to sphingosine. These are relative amounts of activity here, relative amounts of activity. Likewise, ceramide is C1P. C1P, remember, works opposite of ceramide. Again, showing you some relative ratios. Now, let's take a look at the ER, right? So the ER can actually transport ceramide all the way to the plasma membrane, right through the Golgi apparatus. But of course, ceramide can be broken down into CPE, okay? And of course, the de novo, that's ceramide phosphate ester, okay? Or basically ceramide 1-phosphate. Here's the de novo pathway to build ceramide up, and that's its translocation from the endoplasmic reticulum. Remember, too, that sphingosine 1-phosphate can be completely broken down to this DEG product, and I believe that's the end product there. So you got this going on, okay, in the ER. Now, again, what about the lysosome? And ultimately, the membrane itself. In the lysosome, you have all kinds of interesting activity. Sphingomyelin, right, in association with GSL will make ceramide. Ceramide broken down to sphingosine, as we've seen. Sphingosine, phosphorylated sphingosine 1-phosphate, and sphingosine, free sphingosine can also enter the ER, okay? And then that metabolism we just showed you can commence intracellularly from one organelle to another, okay? Likewise, in the membrane itself, sphingomyelin or esters of ceramide phosphate can be metabolized to ceramide, ceramide sphingosine via multiple pathways, okay? So there's recycling and degradation and resynthesis and degradation happening all the time with the sphingolipids, all the major players, including the membrane-associated ones. And there are the enzymes involved. You know, the de novo pathway, the glucosyl ceramide synthase, sphingomyelin synthase, the ceraminidase, etc. All the ones we've talked about multiple times in previous talks. All right. Off with the glasses. Terrible glasses. Sphingosine 1-phosphate improves human ovarian tissue xenografting by promoting angiogenesis and stromal cell proliferation. This was a result of this paper. So far, it was not shown that S1P could protect against chemotherapy-induced ovarian follicle death. Well, this paper is going to resolve that. The work was actually done to evaluate the effects of S1P on human ovarian tissue during exposure to chemotherapeutic drugs like doxorubicin. The results clearly showed, I'm not showing the data because, again, too much molecularity in the talk, so just giving you the summation of the data. We will go through the data carefully, vet it out, and compare the evidence very significantly when we work to help people with their specific research problems or medical issues. The results of this paper anyways demonstrated that S1P protected against chemotherapy-induced loss of fertility. So in the animal model and in cell culture. So this might be very promising and very important for fertility. Now, here's a paper that came out very recently, and it's a review article called The Mediators of Inflammation, published just last year. It talks about our two friends, the topics of today, sphingosine 1-phosphate and ceramide 1-phosphate. If you haven't already figured it out, both of those phospholipids phosphosphingolipids regulate cell growth, survival, migration, immune cell trafficking via cytokine and chemokine synthesis and secretion, angiogenesis, inflammation, and cancer. So major players, right, in pathophysiology and in normal physiology. S1P and C1P are the regulators of sphingolipid metabolic network, right? And since their accumulation causes a net decrease in proapoptotic ceramide, S1P and CIP thus promote pro-survival signaling, okay? So remember, because they build up, you're going to get a net decrease in ceramide, and ceramide is pro-apoptotic. So obviously that couple, that phosphorylation of either ceramide directly or the ceramidase making sphingosine, and then its phosphorylation via any number of sphingosine kinase isoforms, either extracellular or intracellular within the matrix, is going to then modify the relative concentration, steady state concentrations of ceramide itself, thus control apoptosis in the cell fate for that cell system. Okay, by promoting pro-survival signaling. Remember, pro-survival signaling sounds really great, right? No cell death, really important, like say in neurons. Not so good, of course, when you're talking about what? When you're talking about cancer. That's correct. All right, back with these ugly glasses. We've already seen this, but this is, I think I like this picture better. Just talking about where sphingomyelin can be converted to ceramide, to sphingosine. This is happening in the lysosome. The salvage pathway taking sphingosine and rephosphorylating it to S1P. The sphingosine itself can come directly from the nucleus or from the endoplasmic reticulum. Remember that you could also get Denovo Synthesis. That's where it actually occurs, is in the ER. There's where ceramide is synthesized, and ceramide, again, can be transported. That's what the CERT is, ceramide transport to the Golgi. Or remember, after ceramidase makes sphingosine, right? That's all happening with the intraorganellar activity and transport. Remember, or maybe we didn't talk about this time, but sphingomyelin is broken down to ceramide via a neutral sphingomyelinase in the mitochondrion. And then the ceramidase will make sphingosine, and then that will then be phosphorylated, and sphingosine 1-phosphate is involved in respiration and electron transport chain. Okay, it's associated with the regulation of those pathways. Keep in mind, too, that ceramide 1-phosphate can be transported maybe through other organelles, but certainly directly to the plasma membrane. And also the sphingomyelin in the plasma membrane can metabolize to sphingosine 1-phosphate. And as we've been saying, now sphingosine 1-phosphate can leave the cell entirely and can act in autocrine fashion, coming back to its receptor. Right? So a very complex dynamic here. So what about these two lipids and cancer progression? Well, not a good story, sorry. So intracellularly generated S1P, which is secreted out of a cancer cell by any number of transporters, in fact, also ATP binding cassette transporters, or the ABC mega family of transporters. Extracellular S1P is a ligand for G protein coupled receptors, such as the S1P R1 through 5. The intracellular sphingosine 1-phosphate binds and modulates a ubiquitin ligase activity. Mitochondrial S1P binds to proinhibitin and regulates complex 4 assembly and respiration. Remember what I said, how it affects the ETC and OXPHOS? That's where it does, how it does it specifically. Nuclear S1P binds and inhibits histone deacetylase. So if you inhibit histone deacetylase, it means you maintain the acetylation pattern in histones, which means you're going to get a lot of gene expression. And in cancer cells, that's a no good. Epigenetically, of course, regulates histone acylation and transcription of all those genes. And that's called an epigenetic phenomenon because it's the acetylation of the histones with the histone codons associated with covalent modification of DNA as well, such as methylation, acetylation, ubiquitinylation, assimilation. All of that, sphingosine 1-phosphate, then, those kinds of activities in the wrong environment can progress tumors, can cause metastasis, can enhance cell survival of cancers, can cause migration of cancer cells, angiogenesis, inflammation, which is not a good thing in cancer, because out of control inflammation can lead to cell proliferation. Not that inflammation doesn't kill cancer cells, it does, but we're talking about uncontrolled. Chemokine signaling, of course, immune cell trafficking, as we've mentioned, and this epigenetic regulation. Now, what about ceramide 1-phosphate? Wow. Well, the extracellular ceramide 1-phosphate is a ligand, but we still don't know its precise G protein coupled receptor. There's some early reports about them, but not enough to be able to talk about in this review article. It was published just a few months ago. Intracellular ceramide 1-phosphate binds and activates cytoplasmic PLA2 alpha. So phospholipase A2 generates arachidonic acid, if that's the fatty acid in the 2 position of that phospholipid. And that's going to lead to icosanoid production, prostaglandins, for example. And that is a stress response, right? That's an inflammatory response, which is interesting. Intracellular C1P binds to a transport protein and vesicular trafficking and regulates it. What happens here when you do this in a cancer cell? Again, tumor progression, metastasis, cancer cell survival, migration, inflammation, icosanoid synthesis here because of this, and macrophage function. Icosanoids are bad players in cancer cells. All right. So you can look at this as a priming and then a cascade motif, and this is a temporal sequence going from left to right. So first of all, take a look at what happens. Sphingosine 1-phosphate binds to its protein, which is associated with a G protein coupled receptor. We're not sure exactly how, but obviously the signal is transduced to the nucleus, and in so doing, causes the expression of cyclooxygenase isoform 2. It also stabilizes COX-2 mRNA, so you make more of the protein. And remember, too, that you can, via interleukin-1 beta, through that receptor, you activate sphingosine kinase, and sphingosine kinase is going to take sphingosine and make S1P. So a beautiful autocoid system, S1P synthesized via induction via the pro-inflammatory cytokine interleukin-1 beta, making sphingosine 1-phosphate, leaves the cell, comes back into the G protein coupled receptor. This is the priming part, causes COX-2 transcription. Later on, you got a cascade system. You're still binding the interleukin-1 beta. That's still going on. But look what else it does. It also converts ceramide to ceramide 1-phosphate. Okay? And we know that ceramide 1-phosphate works in conjunction with the sphingosine. Because look here. In association with the with ceramide 1-phosphate, you get further arachidonic acid liberation. You get, of course, the expression of the COX-2 with activation because of the stabilization of the messenger RNA. A lot of COX-2, so you get a lot of prostaglandin E2 synthesis. And, of course, that phospholipase A2 itself is translocated and interacts with the ceramide 1-phosphate. So this, again, is all a rumble-tumble cascade system. It's going to lead to more and more and more of these cytokines and all of these inflammatory compounds, which is going to alter then the cell fate substantially. So here it is working with tumor necrosis factor alpha through its receptor. A series of proteins, including the ERK, are going to activate the sphingosine kinase. You're going to take sphingosine and turn it into sphingosine 1-phosphate, again, leaving the cell. Okay. Here the kinase has to be phosphorylate, by the way, if I didn't mention that. Binds to its receptor, interacts with G proteins, any number of which activates these pathways, cyclooxygenase, ERK, NF-kappa B, and also at the same time shuts down apoptosis. So that way it inhibits apoptosis, it induces proliferation, and it causes cancer. All right. Finally, take a look at this. This is talking about now our macrophage assembly. Remember, you have 2 different kinds of macrophages, a family of them, M1 and M2. And remember that globally speaking, you have 2 types of T cells, Th1 and Th2. You also have a Th17. Now look what happens here. In a dendritic cell, when you get activation of the sphingosine kinase, you make a lot of sphingosine 1-phosphate. okay, that binds to its receptor, generating a tissue factor like IL-1B, which then interacts with thrombin, thus inducing again the kinase. So you're making a lot of sphingosine 1-phosphate. You're turning this dendritic cell into a molecular motor to interact with the naive T cell, okay? Because this is all going to be associated with mobilizing this major histocompatibility complex and putting this receptor on the surface of the dendritic cell. So what this all serves to do, sphingosine 1-phosphate promotes increased numbers of Th2 cells, okay? So where are they? There's the Th2 cell. So you have your naive cell. You have the production, because of sphingosine 1-phosphate, of interleukin 4, interleukin 10, and drops interleukin 12. Ups 4 and 10, drops 12. That's an anti-inflammatory one. You're going to turn it into Th2 cell lineage. Okay, and that's Th2 cell is going to act with sphingosine 1-phosphate autocrine, increasing the production of interleukin 4, which again is going to maintain the activation of the Th2 cell, and it's going to block the activation of the Th17 cell, which is normally going to be producing IL-17, 21, 22, and more tumor necrosis factor. Okay. Now, interleukin 4 isn't just leaving the T cells alone. It gets secreted, and it basically induces a B cell to become a plasma cell. The plasma cell, of course, switches from IgM to IgG synthesis, and now you have IgG now recruiting and activating basophils, right? You're going to get degranulation, more interleukin 4 production, right? And you're going to generate mast cells, which are going to in fact interact with basophils, and those basophils with now the plasma cells. So you have a whole turned on acquired immune system, all because of the early synthesis, the activation of the sphingosine kinase pathway. Okay, and likewise, we don't really know yet, but probably the Th1 pathway is turned down, although there are receptors for it. When you bind to the receptor in Th1 cell, it blocks interferon gamma. Okay, so that's going to have an effect on the mobilization of the sphingosine phosphate into that cell lineage. Right? Monocytes, of course, are also going to activate M2 macrophages with the help of sphingosine 1-phosphate, whereas sphingosine 1-phosphate is going to block the activation and the acquired competence of M1 macrophages, okay, when it binds to its receptors. So let's go back here. During sepsis, production of sphingosine 1-phosphate and activation of its receptor in dendritic cells leads to inflammation and coagulation in the blood. Sphingosine 1-phosphate impairs the initiation of Th1 cells, just went through that, by suppressing interleukin 12 production by dendritic cells, but it promotes the Th2 lineage by increasing IL-4 and 10. Activation of the CYP1, as the sphingosine 1-phosphate receptor isoform 1 by its ligand, is activated in T cells and inhibits interferon gamma production. We just saw that, okay? Inhibiting interferon gamma production is the key here. And overexpression of the receptor increases the amount of IL-4 production. Sphingosine 1-phosphate also links to differentiation of the macrophage lineage, going from IL-10 to produce the M2 phenotype. So you're going to increase the M2 phenotype. Now, if you listen to the last two talks on sphingolipids, you know that's really important, and that's how we're going to end this. Because if sphingosine 1-phosphate plays a role in macrophage activation and differentiation, look what that means. Now, M1, macrophage lineage type 1, produce inflammatory cytokines, whereas macrophage family 2 induce low production of the inflammatory, but a high synthesis of the anti-inflammatory. Sphingosine 1-phosphate promotes the type 2. So in the end, you inhibit the inflammatory response. And isn't that what we already know about sphingosine 1-phosphate? It's cell promotive. So although it's working through the inflammatory response, it's basically putting the brakes on of inflammation, while it's proliferating T cell lineages, which can cause further inflammation downstream at tissue types not associated with, for example, with cancers, or even with the resident macrophages that are involved in this pseudo-regulation of M1, M2 differentiation. So that's all I want to say right now. Hopefully that gave you a good overview again of how sphingosine 1-phosphate plays a very crucial role in human health. both at the level of pathophysiology and straight up healthy tissue morphogenesis and activity. Again, we just barely scratched the surface there. I talked a little bit about reproductive biology being associated with sphingosine 1-phosphate metabolism, basically shielding against ceramide. I talked about how sphingosine 1-phosphate can help repair, or that is decrease the amount of tissue damage caused by chemotherapeutics in frank cancer chemotherapy patients, but that at the same level, by the same token, you also can get a proliferation of cancer cells, and you can get a depression of the immune response, which are both bad because that can lead then to tumorigenesis. So again, sphingosine 1-phosphate, major autocrine, paracrine signaling compound, also working directly within the cell synthesized, works at multiple levels, metabolic, signal transduction through canonical pathways, and also very likely involved in gene expression, if nothing else, the expression of its own receptor. Chromatin remodeling, epigenetics. So I really want to thank you again for your undivided, I hope, attention, and that you gained something from this talk. I always try to come up with some classical papers so that we move into an area. You get to feel comfortable about talking about something that sounds at the beginning somewhat exotic and rare, like sphingosine 1-phosphate. And then I try to bring home some very recent papers to let you know what's going on in the field. Today I just gave you the review because I want to go into much more detail about C1P, S1P, and ceramide playing against each other in various types of chemotherapy and immune checkpoint inhibition of cancers, as well as maybe some interesting stories in heart failure research and in other kinds of ischemic stroke and things like that, other things associated with the cardiovascular system. All that literature I know is out there, and I just really enjoy tapping into it, and that's what I hope to do soon. So again, thank you very much for your attention. It's been grand, and please enjoy the rest of your day. 2018 should be a fantastic year for all of us, as long as we believe. So thank you very much for your attention, and goodbye for now.